everybody, welcome to another edition of the Lab 207 webcast. My name is Mr. Kai. I'll be hanging out with you today, and today's a notable day. We finish up our series in energy. This thing's been like 10 videos long. Our last topic for the day is the Calvin cycle of photosynthesis. So let's get our objectives and get going. There are only two of them today. Here's what I need you to know by the end. First one, be able to list the inputs and outputs of the carbon cycle. So what goes in, what do we get out? The second thing you need to be able to do is describe the process involved with the production of sugar. Pretty simple, pretty to the point. Let's talk about it. So an overview first so that you can keep everything straight. Calvin cycle. If we're going to talk about location, this occurs in the stroma. So think about your diagram of a chloroplast. You got a little pancake burger shaped thing. It's got all the green thylakoid membrane stacks on the inside. The fluid surrounding those stacks of thylakoid membrane is the stroma, so the Calvin cycle happens in that fluid. The inputs, the things that go into the Calvin cycle, are ATP and NADPH, also CO2. The ATP and the NADPH come to us from the light reaction. CO2 comes in through the stomata of the leaves from the outside world. Our output of the Calvin cycle is G3P, which is a sugar. That sugar will later be used to make other carbohydrates inside of that plant. And the final thing, and this is probably the most important thing, it does not require light. All the energy needed to run the Calvin cycle comes from that ATP and the NADPH that we get from the light reactions. In the Calvin cycle, there are three stages to be aware of. The first stage is carbon fixation, so that's the piece where we are taking in the carbon dioxide and we're rearranging it into a form that can be used to build sugar. The second part is reduction, so that is the piece where you are actually taking those carbon-containing molecules and building them into our G3P sugar. And finally, regeneration of RuBP. He is the molecule that starts off this cycle. So since he's a starter and it's a cycle, we've got to regenerate him. So without further ado, here's the Calvin cycle, and I'm betting this is going to be pretty quick. So let me turn on my pen and we'll notate a couple things. See up here at the top, we've got a little diagram of where all this is happening. So a couple things to note. We've got light coming in, light running the light reactions in our thylakoid membrane. Water's coming in, and they're showing you in these light reactions the output is oxygen, and we've got ATP, which you can barely see, and NADPH. That NADPH and ATP is going to the Calvin cycle, which is happening in the stroma. The Calvin cycle sends NADP plus and ADP back to the light reaction, so that we got a continuous cycle going on. Into the Calvin cycle, we've got carbon dioxide coming in, spinning through the cycle, and sugar is coming out on the other side. So here's a little more detailed view. When you take biochemistry in college way back in the day, or not way back in the day, way forward in the day when you get to college, you're going to have to memorize all these little steps and the enzymes that get them there, but for our purposes, you don't have to know them all. So here's the basic stuff you need to know. First thing, input. Our input is CO2. That CO2 is bound to a protein called rubisco. He is our first step in this process. When the CO2 binds to the rubisco, that is carbon fixation. The carbon is now part of the cycle, and it can continue on its merry little way. So our molecule that is formed gets over to the second stage right here, which is reduction. In this phase, reduction, we've got our molecule going through a bunch of different intermediates. As it's going through its intermediates, it is picking up electrons and atoms all the way through that process. So if you remember, reduction is essentially the charge of a molecule being reduced as it adds electrons and as it adds different atoms to it. That's what's happening here. Our molecule is being reduced all the way until we get to a sugar, G3P. Now, most people think or say that the Calvin cycle spits out glucose. That's not quite right. This G3P is a sugar that when you attach two of them together, actually when you attach 6 of them together, you get glucose. So when we are talking about monomers and polymers, our monomer of a carbohydrate or a polysaccharide is a monosaccharide. This cycle spits out the monosaccharide G3P that will then later be used to make glucose and all the other sugars. Now that we've got our sugar made, we need to regenerate rubisco since this is a cycle. So We are going to go from our G3P through a couple intermediates. We're going to use up some more ATP, and finally we'll get back to Rubisco. Now that Rubisco is rebuilt, he can go through the cycle all over again. So just a couple takeaways. Now your input CO2, it gets fixed, goes through this whole cycle, is consuming quite a bit of ATP, and it's also consuming NADPH, and out the other end we get the sugar G3P. Let's finish up this whole series with a quick recap of photosynthesis. So in the light reactions, they occur in the thylakoid membrane, they receive light and water, and they give off oxygen, thanks for us, ATP, and NADPH. Our dark reactions happen in the stroma. They receive ATP and NADPH and CO2, and they produce the sugar G3P. Also know the light reactions are known as the light-dependent reactions because they cannot happen unless there is light presence. So I'm going to breathe a sigh of relief. You breathe a sigh of relief. That is the end of our energy series. I hope that you found it helpful and maybe a little enjoyable as online lectures can be. So when you get a chance, join us again on the Lab 207 webcast. My name is Mr. Kite. We'll see you again.